Hello everyone, my name is Esmeralda Escobar Muciño. I'm postdoctoral student from IPC Mexico. Today I'm presenting the work was named Bioinformatic Search and in silico study of the Diels alderase enzyme and transcriptional activator related to the synthesis of the cuautepestalorin in the genome assembly of Pestalotiopsis SPIQ11. Pestalotiopsis is a pathogenic fungus of fruits that causes losses in agriculture. Rivera Chávez in 2019 discovered a new compound produced by Pestalotiopsis sp. IQ11, known as cuautepestalorin, whose biological function is similar to acarbose, a drug used as a treatment for type 2 diabetes. Cuautepestalorin is produced by a Diels-Alder reaction using cytosporin M and oxopestalopromane as substrates. The aim of this work was to assemble the genome of Pestalotiopsis SPIQ11 to identify the participation of these alderase enzymes in the production of cuautepestalorin using molecular docking screening. Using a genomic protein blast pit, 38 disalderase enzyme sequences were found which were compared with the MALC template protein from the fungus Malvaceae aurantica. In this picture, the 38 genomic contexts of the disalderase are drawn. Also, 4 types of genomic contexts related to the disalderase were found. On the other hand, each of the 38 proteins were modeled on the Swiss model server using the Malvaceae aurantica enzyme MALC as a template. In each of the 38 enzymes, the catalytic and NADPH binding domains were identified. The molecular docking screening was made with the 38 disalderase with the putative substrates of the cuautepestalorin synthesis pathway. The software used for the molecular docking screening was MOE, obtained the Gibbs energy of each interaction. Showed a range of Gibbs energy from -5.7 to -8.4, with 3 interactions considered the best. In this picture are shown the best 3 results of the molecular docking screening when interacting with the substrates and cofactor NADPH. The conclusion of this work was divided in 3. The first was the bioinformatic tools helped to assemble the genome of Pestalotiopsis SPIQ11. The second was the 38 disalderase enzymes were found in the genome of Pestalotiopsis SPIQ11. Finally, by molecular docking screening, it was possible to find the SDR family as the best ligand interaction, which serves as a starting point for a heterologous cloning analysis to quantify cuautepestalorin.